This is part 3 of the cell communication lecture. Make sure, this is just kind of a side note, but make sure that you go over the study plan on Moodle for this chapter. The slides are a little bit um, lacking. The slideshow is very short compared with the amount of information that's in this study plan. So make sure that you go through that study plan. The slideshow can kind of introduce the topics, but you need to do the study plan and the questions within the study plan to really understand it. Okay. Depending on the status of the organism, cells can respond by storing energy as glycogen or fat, or making it available in the form of glucose. So basically, glycogen is, well, this is going to represent one glucose molecule. This is one glucose molecule, and then glycogen would be many, many chains. Oh, that's not right. Hold on. Okay, so be many, many chains of glucose. So when our liver cells or our muscle cells want to remove glucose from the blood and store it, they store glucose as glycogen. And then when there's excess glucose and the glycogen stores are full, that excess glucose can be converted to fat. That's the reason why they say sugar makes you fat. Um, that's not exactly true, but excess sugar does. So a signal transduction pathway allows muscle cells to respond to immediate requirements for energy in the form of glucose. So it allows these muscle cells to break down glycogen and remove glucose from the glycogen, or remove glucose from the blood and store it as glycogen, depending on what is needed. And liver cells can do the same thing. Cell growth is almost always stimulated by external signals called growth factors. I think one that we already discussed was epidermal growth factor. Uncontrolled cell growth leads to cancer. Uncontrolled mitosis or uncontrolled cell growth leads to cancer. And mutations in the genes encoding protein components of signaling pathways are often found in tumor cells. So when we go back to these different signaling pathways, this one, for example, epidermal growth factor has to bind, actually two epidermal growth factor ligands have to bind to two epidermal growth factor receptors, here and here. And when both of those events happen, this pathway, I won't name all these molecules, but this pathway where we've got the RAS attached to the GTP, well, this complex, RAS GTP, phosphorylates RAF, and RAF phosphorylates MEK, and then MEK phosphorylates ERK, and that ERK that's phosphorylated can now enter the nucleus and attach to the DNA and stimulate genes that cause the cell to grow, to migrate, to adhere to other cells, angiogenesis, which is the growth of new blood vessels, and it can also inhibit apoptosis. And this is great if we want our cells to grow, but what if this is a cancerous cell? If it's a cancerous cell, it could cause it to grow indefinitely into a tumor. And here is another example of a pathway dealing with phosphorylation. Um, excuse me. ERK phosphorylates MNK1, and then MNK1 phosphorylates ELF4E. Uh-oh. Sorry, I'm having some technical difficulties. Okay, so the MNK1 phosphorylates ELF4E, which is associated with messenger RNA, and it signals the phosphorylated ELF4E signals the messenger RNA to begin transcription, or the process of making a protein. Now, cell death, when it is programmed into the genetics of the cell, we call it apoptosis. And apoptosis is important for removing damaged or unnecessary cells. The use of cellular signaling to organize the dismantling of a cell ensures that harmful molecules from the cytoplasm are not released into the spaces between cells, as they are in uncontrolled death or necrosis. Apoptosis also ensures the efficient recycling of the components of the dead cell. Termination of the cellular signaling cascade is very important so that the response to a signal is appropriate in timing and intensity. Degradation of signaling molecules and dephosphorylation of phosphorylated intermediates of the pathway by phosphatases are two ways to terminate signals within the cell. Let's go back to, I think it's this one. Okay, so let's say that the GTP ACE, which I don't know if you've caught on, but anytime it ends with ACE, it's an enzyme. The GTP ACE is inhibited. So the GTP ACE does not break down this GTP. So what's going to happen is this cell is going to continue to grow and grow and grow and grow. It's never going to stop. With the presence of GTP ACE, then there's going to be a limit on how much growing this cell does. But if GTP ACE is inhibited somehow or mutated, then this cascade of reactions is just going to happen over and over and over and over, and that cell is going to multiply many, many times, which is cancer, basically. Okay, these are yeast cells under a microscope, and this would be a single yeast cell, and this would be a yeast cell that is producing another yeast cell by budding, and same thing here. Yeast are eukaryotes. That means that their cells have a nucleus, but they are single-celled fungi. They are single-celled fungi. We use yeast to make bread and beer, and the signaling processes are similar to those of multicellular organisms. Budding yeast are able to participate in a process, and this is budding. These are budding yeast. This one and the other one as well. In a process that's similar to sexual reproduction that entails two haploid cells, that means cells that have only half the normal number of chromosomes, combining to form a diploid cell, which has two sets of chromosomes. It would be like the haploid sperm in a human has 23 chromosomes, and the haploid egg in a human has 23 chromosomes. And when they combine, they form a cell called a zygote that has 46. Only this is happening in yeast, not sperm and egg. It's happening in haploid yeast cells. Um, and when they combine, they form a diploid yeast cell, one that has two sets of each chromosome. In order to find another haploid yeast cell that is prepared to mate, budding yeast secrete a signal molecule called mating factor. So if this one secretes mating factor, then it can combine with this one, and they can unite and form a diploid yeast cell, or a yeast cell that contains two copies of each chromosome. In bacteria, the first evidence of bacterial communication was observed in a bacterium that has a symbiotic relationship. That means it lives in a close association with Hawaiian bobtail squid. When the population density of the bacteria reaches a certain level, specific gene expression is initiated, and the bacteria produce bioluminescent proteins that emit light. Because the number of cells present in the environment, that's basically what cell density is the determining factor for signaling, bacterial signaling was named quorum sensing. Autoinducers are signaling molecules or ligands secreted by bacteria to communicate with other bacteria of the same kind. The secreted autoinducers can be small, hydrophobic molecules, which can pass directly through the phospholipid bilayer, or they can be large peptide-based molecules that must bind to a receptor on the cell surface. When AHL enters target bacteria, it binds to transcription factors, which then switch gene expression on. Okay, so if we see here, there's low cell density and the genes are regulated by autoinducers. That's what we're looking at right here. These are the genes, and a bacterial chromosome is circular. The receptors look like little triangles, and the autoinducers are the red dots. So when that autoinducer is released and diffuses away from the cell, when the cell density is low, it's not going to have anything else to bind to. But when the cell density is high, more autoinducers are present. You've got many, many more autoinducers present. They bind to receptors that regulate the transcription of certain genes. So by binding to these internal receptors, and you see how they've done in all of these different bacteria, they can then regulate these genes on the bacterial chromosome, and the genes responsible for the production of autoinducers are expressed. And so it's a positive feedback loop. In other words, the greater the number of cells, the higher the cell density, the greater the number of autoinducers. I'm just going to put auto. And then that's going to increase the cell density, which is going to increase the number of autoinducers. That's a positive feedback loop. On and on and on until the cell density is so high that the bacteria release that phosphorescent bioluminescent glow. All right, what are the different types of signals? We have the intercellular and intracellular, either between different cells or within the cell itself. And the signals can be paracrine, which means that